want to turn our attention from the general retinotactile system to a specific system with a very specific function. I want to turn our attention now to the oculomotor circuit for saccade control. So we'll first want to talk about the difference between saccades and eye and head movements. Then we'll have to notice that there isn't just one thing, the saccade. They come in different sizes, occur at different frequencies, and so on. The process of saccading includes saccade initiation, fixation in the saccaded area, and then an inhibition of return to a previous location. We'll want to figure out what the general mechanisms are for saccade initiation. How does the brain control those saccades, move the eyes around as it does? And finally, how does it figure out what it wants to saccade to? How does it decide the important parts of the visual field? So I've shown you the picture on the left many times. It's a painting. On top of that painting, those white lines are a trace of the saccades of a person when they're viewing that painting. And there are two important things we'll want to notice here, right? One is that certain areas have a very high priority. They're saccaded to a lot. Others, not so much. And in addition to that, there is a sort of general line. You don't see a lot of backtracking in saccades, going back to the same place. So all of these are movements of the eyes. They're movements of the eyes that aren't following anything and that aren't the result of head movements. So our saccades are just movements of our eyes that occur relatively autonomously from any particular visual stimulus that might catch our attention. So when we have, for instance, smooth eye pursuit, when we're following something in the visual field that's moving, right? We move our eyes in this direction, but that's different from saccading. We might even move our head to turn to follow it. That also different from saccading. Saccades very specifically are movements of the eye that direct that fovea, that highly refined processing stream that's so very small, right, towards particular areas in the visual scene to do that fine-grained processing in that area. Now, we've generally talked about saccades as though there was just one kind, but in fact there are a number of different kinds of saccades. There are major saccades and minor saccades. Within the major saccades, there are two groups. Large saccades, which are relatively large movements across the visual field. Those tend to occur not that frequently, every 3 to 20 seconds. They're not studied nearly as intensely as the other major saccade, the small saccades. These are the ones that we normally think of as saccades. They're movements of the eyes, relatively small in terms of degrees, right? But nevertheless, they occur about 3 times a second, and they're the ones that really seem to be the high-level processing of individual features in the visual field for the purposes of our visual processing. There are also minor saccades, though, and they fall into 2 classes. Mini saccades are flicks, and microsaccades are tremors. These are just little movements, right? Little tremblings of the eyes. Small saccades, mini saccades, and microsaccades, generally speaking, can't even be detected without special apparatus. So we want to keep all of those things in mind, and we also want to keep in mind how saccades occur. There's an initial saccade initiation. There's a fixation on the saccaded area, and then there's an inhibition of return, so that saccades are forever moving forward from one to the next, initiating, fixating, going to the next spot, and not just going back and forth, back and forth, back and forth. So how does the brain decide what it wants to saccade to? What drives visual attention? Well, theories of visual attention hypothesize one or more salience maps. Salience being importance, significance. And these maps direct attention to particular areas in a visual scene. They determine what's saccade worthy, as it were. Now, some cortical cells are known to encode saliency. In addition to that, though, these superficial visual layer neurons of the superior colliculus, they function to encode a visual saliency map when scanning the visual scene. The diagram on the left here depicts one model of this process from this article by White, where I pulled the diagram. So we have our occipital lobe, our parietal frontal, our thalamus, our brainstem, which controls the actual motions in our eye. So the work by White and colleagues suggests that the superior colliculus actually encodes saliency and priority and uses it via the brainstem to drive saccades. So it looks like that superior colliculus system functions importantly in prioritizing and determining where it is that we devote those important resources, even though it isn't a part of a full-blown object recognition system like we find in the visual cortex and its projection down into the inferior temporal lobe.